Hi, everyone. My name is Sathvik. I'm a senior at Amador Valley High School, and my project this season was the detection of autism spectrum disorder, or ASD, as I'll refer to it for the rest of this presentation, through the analysis of brainwaves using machine learning. And I worked with Pragati Dharmale under the Department of Computer Science and Engineering at ASDRP. And, excuse me. So this is the abstract. I know most of you can read it, but some of you might be auditory listeners or learners. So autism is a developmental and neurological condition that is historically difficult to diagnose due to it being a common within a spectrum. The symptoms are not defined or discrete. And due to that, we need an unambiguous analysis of symptoms to diagnose a patient very well. So our model, which the use of the 1020 system to isolate channels and the extraction of data and using pandas data frames, that all creates a model using Python to help doctors diagnose in a more accurate and efficient way. So to those of you that don't know what autism spectrum disorder is, or ASD, it's a brain developmental condition, as I've stated before, and it controls basically your entire life in communication, behavior, and social interaction. And it often co-occurs with other disorders like ADHD and other genetic syndromes. And why we're doing this is because the early prediction of ASD and the early intervention can be extremely beneficial to patients and doctors alike. And the current tests, like I've said before, they lack predefined tests to diagnose. So this project would help the doctors. And our objectives for this project were to study the anatomy of the human brain and to analyze EEG signals, which I'll get into more depth later, and to develop algorithms to detect the ASD and to compare and analyze different algorithms based on performance and then validate them later. We'll move on. So EEG, or electroencephalogram, is a brainwave. Basically, it's how you collect brainwaves. And there are two types. It can be either non-invasive or invasive. Our project is focusing on non-invasive, so you won't have like syringes or cords going into your head. And it is the electrophysiological monitoring technique that records electrical activities of the brain over the scalp using tiny little circles that we call electrodes. And the activity is really tiny. It's measured in microvolts, so it's really, really, really tiny. And this is a quick block representation that you can see, which is basically what I stated in the last slide. And there are 4 different types of EEG waves, delta, theta, alpha, and beta. They're classified as below, and this is what you'd see. This picture right here is what you'd see when you'd graph the data that we collected from our patients using a headset called the Emotive headset. Right now, that is not FDA approved yet. We're waiting on that, but it's still safe to use, and we use that to collect our data through delta, theta, alpha, and beta waves, and we use mostly all of those in our project. And we use the 1020 system to identify where to place electrodes, or those little circle things, to collect data. And your brain, basically, in terms of data collection, is split into 4 different parts: the nasion, the inion on the back of your head, the left preauricular point, and the right preauricular point. And with a non-invasive headset, you get 14 possible channels, as shown in this bottom left image right here. And we use 7 of those. No, 8 of those. I can count. And those were F7, F3, P7, O1, and O2, P8, and AF4, which you'll see a lot of later. And those different channels gave us different waves.
Questions are still free.
Yeah. Yeah, we used all 3 of these different, all 8 of these different channels to get our data. And then furthermore, we used 3 different filters to separate and classify and analyze our data. We used the low pass filter, which allowed low frequencies to go through, high pass filter that allowed high frequencies to go through, and notch filters, which allowed certain frequencies to go through. And the different Python libraries we used were SciPy, NumPy, Seaborn, NetworkX, Matplotlib, Community. And furthermore, we separated all the data we got into 3 different sections. data that was collected when a patient was focused, unfocused, and drowsy. And in the next slide, our results show that we used a correlation matrix to determine correlation between all of those different channels. So since each of those different channels from your brain and the non-invasive headset gave you data, we used those to compile a matrix to determine which channels had correlations with each other. And these correlation matrices were then used to create heat maps, as seen in the right. And these heat maps basically show which channels are interfering with each other. So in unfocused, which is top right here, you can see that the F3 channel is interfering quite a bit with P8. So that just tells us what we need to refine and work on more. And in conclusion, our project was pretty early in a stage of development. There's a lot to learn from and grow from, and our project hasn't, well, since it's in the starting stages, we haven't really got that far. With efficient uses of time, resources, and knowledge, and recognizing ASD and its symptoms early could be extremely beneficial and helpful to various patients across the globe. And I've gotten a lot more exposed to the Python language and libraries that I honestly didn't know existed. And EEG as a concept was entirely new to me, so learning about it was extremely productive and informative. I would like to thank my advisor, Pragati Dharmale, for advising me this season, and her coworker, Mishesh. Without their help, the project would have been basically impossible. And I would also like to thank all of ASDRP for creating such an educationally nurturing environment. And these are my citations. I'll leave. Oh, we are at 20 right now, but if you have any questions, feel free to drop them in chat. Thank you.